Good morning, everyone, and welcome to my talk. Thank you for coming. Today I'm going to tell you about two species of a weedy annual plant that commonly hybridize. So first, I want to talk about what is hybridization and why should we study it. Hybridization, or interbreeding across genetically distinct lineages, can be frequently observed in many groups of organisms, including animals, bacteria, fungi, and plants. It's especially common in fish and in flowering plants, and is estimated to occur in up to 25% of angiosperm species. So hybridization is an important and interesting process to study because it impacts the evolutionary trajectory of the lineages involved, and it influences genetic diversity. And every hybridizing system that we study tells a unique story regarding the mechanisms by which hybridization takes place, and as well as the outcomes of hybridization between lineages. And so now we have some background on hybridization. I want to tell you about hybrid zone formation and processes. So first, before a hybrid zone can form, there is uninterrupted gene flow between two groups. And here they're represented as two lineages, each with three individuals. And also a hybrid zone is an area where hybridization simply occurs between two groups. So second, it's possible that a barrier to gene flow can form between these two lineages, isolating them, and this can be temporal, functional, or spatial, or a combination of those types of barriers. Next, these isolated lineages can diverge due to exposure to different conditions, favoring different traits, or random genetic drift. And then one possible outcome of this process is active hybrid zone formation. If these two diverged lineages come into contact again and the sympatric barriers are not strong enough to prevent gene flow between them, an active hybrid zone can form, and this is what I have been investigating in my study. Once the hybrid zone is active, it can have a few potential outcomes, including reinforcement of boundaries between species, or between lineages, where hybrid formation becomes more rare and the lineages continue to diverge further, or stability can happen, where an active hybrid zone is maintained. However, in biological systems, this is a lot more complex, and there can be evidence supporting stability and reinforcement at the same time, and it's not always clear which of the outcomes is happening, or the line between these two outcomes is not always clear. So what potentially occurs in one of these hybrid zones? This is just a simple example showing one trait, which is flower color, and how hybridization could potentially affect the phenotypes of individuals. In some cases, F1 hybrids show intermediate trait values. They can also retain the trait values from either parent, but this just depends on patterns of genetic inheritance. If F1 hybrids are intermediate and fertile, F2 hybrids can form. And they're usually more diverse in this phenotype, and can exhibit extreme phenotypes, even more extreme than either parent. And then after many generations of back crossing, you can sometimes observe a gradient of intermediate phenotypes in these hybrids. So I'm interested in exploring these patterns in my study system, which is Triodanis. And this is a common weedy annual plant in the Campanulaceae family. There's currently 7 described species. It's commonly found in relatively disturbed areas, and the whole genus produces open purple flowers as well as closed oblically selfing flowers. And importantly, several of these species are known to hybridize, and our lab members have observed intermediate phenotypes between the two species in the field every season for the past decade. So this one on the left is Triodanis biflora, and the one still on the left but more towards the middle is Triodanis perfoliata. And these are the two focal species of my study. They have important morphological differences, which we'll get into, but they were selected not only because they're cute and beautiful, as you can see here, but because they hybridize very extensively. So there's plenty of opportunity for hybridization between these species. These two species overlap greatly in their ranges, and the range overlap is indicated by the muted yellow color there, whereas the range where only Triodanis perfoliata is found is in the blue. And so also when they co-occur, we know that they tend to co-flower, which we have also observed in the field. And this study by Colette Berg, Burr, and Weber shows this overlap in flowering time. So based on this, there's plenty of opportunity for hybrid zone formation, both spatially and temporally. The other reason I chose this study system is that past work has consistently reported intermediate phenotypes and has suggested that hybridization is occurring in the system. In this figure from Bradley's hybridization paper, leaf width and capsule pore position here on the left is more typical of Triodanis biflora, while those on the right are more typical of Triodanis perfoliata. And then those in the middle are intermediate phenotypes, and hybridization is the most likely explanation for these intermediates. In addition, Carol Goodwillie and Emily Rose Stewart also provided evidence that hybridization is occurring in Triodanis. And both of these studies have confirmed the presence of intermediate phenotypes by hand crosses between the two species. So we see these patterns of intermediacy, but yet Triodanis biflora and perfoliata still form distinct monophyletic groups. Ongoing work by Taylor Simmons, a PhD candidate in the Weber lab, indicates this and shows monophyly. So these two species form monophyletic groups with strong support represented by many populations. And this is where I come in. Based on these species forming distinct groups, despite so many intermediate morphologies, I would like to ask the following questions, which are first, I plan to provide a genetic signature of hybridization. Second, to determine if morphology can be used to distinguish Triodanis biflora, perfoliata, and their hybrids within hybrid zones, and also characterize patterns of morphology across hybrid zones. And third, I would like to address the question of what factors could be working to maintain the species boundary. So no previous study has presented a genetic signature of hybridization in this group. So unfortunately, we were unable to use samples from my field season because that was too recent, but that is a future project that I've collected tissue samples for. The work this plot that I made was from a RADseq data set that Dr. Weber helped me data mine from previous lab work. And the DAPC plot, which is basically a PCA for genetic data, that shows clustering of both parental groups and their hybrids separately, regardless of their state of origin. This includes biflora, perfoliata, and hybrid individuals, all from the same site in Texas, as well as some biflora individuals from Missouri and some perfoliata individuals from Kansas. And also, this supports our morphology-based determinations that are made in field studies. And also for those more familiar with structure analysis, structure revealed the same clustering pattern shown here. So now that we have a genetic signature for hybridization in this system, I next want to investigate patterns of morphology across hybrid zones. And then finally, potential factors maintaining the species boundary. To address the next two research questions, I investigated several hybrid zones to capture as much variation as possible. I visited 5 hybrid zone sites across the Midwest ranging from Oklahoma through Arkansas up through Tennessee, Kentucky, and of course here in Southern Illinois. And these hybrid zones varied highly in their size and composition. So hybrid zones going from one parental group to another, as shown on the left here, are more experimentally ideal. And some of the hybrid zone sites looked more like this. However, some other sites looked more like the one on the right here, where everything was more mixed, facially intermixed. So to deal with this, I just simply ran transect from the highest abundance of one parent to the highest abundance of another parent and made note of what individuals putatively look like throughout the transect. So to not overcomplicate things, I'm going to stick with the first example for how I ran my transect and method of sampling. After mapping out the site and writing observations, we ran a transect in a straight line between the two parental populations, flagging each meter. We took GPS points at the start and end of each transect and collected 2 to 3 individuals closest to the center line of each meter mark. And these were collected without regard for the species determination. And soil samples and light meter readings were collected at 3 locations along the transect. They were collected at the meter marks where the most individuals of each group were present. And pollinator visitation rates were also taken from these locations, but only for Triodanis biflora and Perfoliata due to only a few of us being in the field. Here's some fieldwork pictures. I was kind of bad at taking good fieldwork pictures, but this gives an idea of what it looks like in the field. And I'd like to give special thanks to Sarah Lukowski and Lady Laura Arias Martinez for their work with their help with my fieldwork. Definitely could not have done this without them. After fieldwork was done, morphological data of 23 quantitative traits was collected from 262 individuals across 7 transects, 2 separate parental populations, and a few individuals that had genetic data associated with them from each group. So here we measured things like leaf width, the total stem length, basal stem diameter, kind of the number of capsules, and a lot more. And I would like to give special thanks again to Brooke Catlett and Kaylee White, who helped a lot with these measurements. Would not have gotten through 262 individuals without their help. So to investigate all the morphological data that we worked so hard to collect, multivariate analysis of variance, or MANOVA, was used to maximize discriminability between groups. And here, the overall model was highly significant, indicating consistent morphological differences are present between these groups across all sites that we investigated. And in the canonical plot, you can see that both parental groups, which is Triodanis biflora and Perfoliata, are almost entirely separated and distinct based on morphology, where hybrids are intermediate and more variable and show some morphological overlap with both parental groups. So even though there are consistent morphological differences between these groups, patterns of morphology varied across sites, likely due to difference in site composition. And so these are principal component analysis plots of 2 example sites, one from Arkansas and one from here in Illinois. And so in these examples, we can see how differences in parental abundance can affect the degree of intermediacy of putative hybrids. At sites where the parental abundances are more equal, which is here on the right, hybrids seem to be more intermediate or equally overlapping with both parental groups. Versus sites where biflora was far more abundant than Triodanis perfoliata, the hybrids overlap more with biflora than they do with Perfoliata. So differing abundance of parental groups could lead to asymmetrical hybridization. Now, I also want to discuss what traits were consistently important for distinguishing between groups. So I found that leaf width, clasping, and the total number of closed flowers were the traits that differed significantly between all three groups. And these were the most important traits of those investigated in our study for distinguishing between groups. These are consistent with taxonomic keys, previous studies, and our own morphology-based determinations. So here I show leaf width as one example. Triodanis biflora leaves on the left are more narrow and elliptic in shape, while Triodanis perfoliata leaves on the right are wider and much more ovate. And then hybrids show intermediate values, but are much more variable in their leaf width as well. So now we've investigated patterns of morphology and determined important traits for distinguishing between these groups. So this brings us to the final question of what potential factors could be working to maintain the species boundary, despite the extensive hybridization shown between groups. And in this study, I investigate potential microhabitat differences in soil texture and light intensity, differences in visitation rates, and differences in breeding system between the species. So I first chose to investigate potential microhabitat differences in light intensity and soil texture because a previous study by Colbert and Goodwillie found significant differences in soil texture, soil chemistry, and light environments between populations of Triodanis biflora and Perfoliata. Shown in the example here, they found Perfoliata soil to have a higher sand content than biflora soil, and therefore biflora soil had higher water retention on average, as you can see by the darker bars being taller than the lighter ones over time. And in the same study, they also reported that Triodanis biflora was found in populations that was found in more sunny areas with only 15% canopy cover on average, while Perfoliata was found in areas with more shade with up to 53% average canopy cover. So in my study, there was a significant site effect and a significant site by group interaction, meaning that at some sites, Triodanis biflora had lower sand content than Perfoliata, for example, but at other sites, it would have had higher sand content in its soil. However, I found no consistent significant differences between groups for soil texture or light intensity. This suggests that once these two species are already co-occurring in hybrid zones, the soil texture and light intensity are likely not representative of significant barriers to hybridization. The next potential barrier I investigated involved pollinators. Kate Tillotson's previous work has shown that pollinator community composition does not differ between Triodanis biflora, Perfoliata, or their hybrids when they co-occur. So I wanted to follow up and assess visitation rates. In my study, I found a significant side effect and significant site-by-type interaction again, but no consistent overall effect between groups in terms of visitation rates. As you can see here, at some sites, the visitation of biflora was much higher, and at some sites, the visitation of Perfoliata was higher. So pollinator visitation is not likely a significant barrier between these species either. This is the mean number of visits over an hour period, but overall visitation rates were very low as well. So it was difficult to tease these two species apart. So now we've investigated microhabitat differences in soil texture and light intensity, as well as pollinator visitation as potential barriers between the two species. And we have no significant differences to show for it yet. But I have one last exciting potential barrier to show you, and that is the breeding system of Triodanis. So Triodanis displays dimorphic cleistogamy, which is a breeding system involving production of cleistogamous or closed ovigately selfing flowers, as well as chasmogamous or open flowers that have the potential to outcross. These open and closed flowers are produced in the same plant um, in all members of the genus. So in addition to the morphological differences I mentioned before, there are consistent differences in chasmogamous flower production between Triodanis biflora and perfoliata. It has been noted in previous studies and in taxonomic keys, and also observed in the field that biflora almost always produces only 1 to 2 open flowers per stem, while perfoliata can produce many more and more synchronous open flowers. This opens up the opportunity for asymmetrical gene flow between species. But what happens in hybrid zones when hybrids with intermediate and variable morphologies are thrown into the mix? In this study, I will first note that we quantified the breeding system as a ratio of the chasmogamous flowers to the total number of flowers, and this is called PCH, which is plotted on the y-axis there. And I found a significant side effect, a significant group effect, and a significant interaction in terms of breeding system. So the group effect means that across all hybrid zones, these groups consistently differ in their allocation to outcrossing. Hybrid allocation to outcrossing falls somewhere in between the two parents generally, but again is more variable and can likely be skewed towards whichever population is more abundant in a hybrid zone or represents the majority of chasmogamous flowers. In addition, the breeding system is likely both genetically and plastically controlled, changing with environmental influence since there was a significant side effect as well. So you may be wondering what these data look like across an individual hybrid zone, and it's pretty cool. So I'm using one transect from Tennessee as an example here. Results in the individual transects reflect the overall pattern, which was found in the original overall bar graph. But the parentals, which are spatially closer to putative hybrids, seem to influence the hybrids that are near them in terms of the breeding system. So as you can see, the putative hybrids that are close to biflora or within the biflora population have a lower proportion of chasmogamous flowers than those that are up closer to the perfoliata population. So in a natural setting, this indicates potential limits to hybrid gene flow if hybrids closer to biflora are producing fewer open flowers. And as breeding system changes, potential for hybridization also changes drastically. So we found that the breeding system to be the only thing of those that we investigated that consistently differed across or between all groups, and that can influence reproductive isolation. This difference in breeding system imposes limits on hybridization, but at all of our sites, we still consistently found intermediate phenotypes that are likely hybrids, and this suggests that stable hybrid zones are likely occurring between Triodanis biflora and perfoliata. So in conclusion, there is a genetic signature of hybridization in between Triodanis biflora and perfoliata. These groups can be distinguished based on morphology. So morphological patterns vary across hybrid zones, but these two groups can be distinguished based on several morphological traits, even within hybrid zones. And some traits could potentially be used to distinguish hybrids as well from both parental groups, but the intermediacy and high variability of these hybrids makes this hard. And finally, the breeding system of Triodanis is likely the main factor that's maintaining the species boundary between the two species by imposing limits on hybridization. So some future Triodanis hybridization projects that could be studied are studying genetic variation across hybrid zones, which I've collected tissue samples for in tubes. And this is important to confirm that hybrids are intermediate to the two species, or if they're more similar to one species or another, and whether these patterns of similarity are related to the parental abundances at different sites. Investigating other potential reproductive isolating factors, such as differential pollen tube growth rates, rates of abortion of hybrid zygotes, soil chemistry, and other microsite variation is another potential future project. Since there's so many potential barriers, I could not look at all of them in one study. So in addition, more rigorous study of hybrid zones and the conditions associated with hybrid zone formation are necessary. In the beginning, I told you that hybridization is common, and each study system represents a unique example of the processes and outcomes of hybridization. And I'll assert again that hybridization is not an anomaly. Many systems hybridize, but many studies investigate taxa and neglect the genetic and phenotypic variation that is present in the hybrid forms between them. So hybrid zones are dynamic and variable within and between study systems. So investigating natural patterns of hybridization, as well as parental as well as factors that affect hybridization and the effects that these have on species relationships in every study system is important to our overall understanding of ecological and evolutionary processes. So studies like mine are important for understanding these naturally occurring patterns of hybridization that occur in many taxa. And with that, I'd like to thank the Illinois Native Plant Society for partial funding to support this project. Dr. Weber for managing to keep me on track throughout the whole endeavor and always being there to help and support me. My committee members, Dr. Newbig and Ed Heist, for their thoughtful suggestions and help. All of the Weber lab members, especially Sarah and Lady and Brooke and Kaylee for helping me so much with my research and their tireless effort measuring so many plants. And then, of course, Taylor Simmons as well for helping answer a lot of questions along the way. And thank you all for coming.